Good afternoon. Um, my name is Dr. Macarena Farcu, and I'm an assistant professor and extension specialist at the Department of Plant Sciences and Landscape Architecture at the University of Maryland, College Park. First of all, I want to thank you all for joining the talk and the interest on the topic. Today, I'm going to be covering the topic of what is fruit quality and the factors that affect it. So let's get an general overview of what I want to share with you today. I will start, of course, talking about what is fruit quality, its components, and how do we measure each one of them in order to get a quantitative value from them. We are going to look at ways to instrumentally measure each one of the components of fruit quality, as well as how do we measure it from a consumer perception perspective in order to correlate these measurements. And then we are going to touch on different types of fruits which are classified differently according to their ripening types or ripening behaviors. We are going to look at what is a climacteric and a non-climacteric fruits. And once we identify their differences, we are going to look as a widely commercially used strategy in order to control ethylene, one of the most important ripening related hormones. And how can this help in order to reduce fruit waste and fruit loss while maintaining fruit quality of fruits? Okay, so with that, let's move forward, and we are going to start looking at what is fruit quality. Really, we know that fruit quality is going to be affected or is going to be composed by changes in color of the fruits. As you can see in these pictures, we have a change of color in apple fruits. We can talk about two different components of color. We can talk about the background color in apples that is still this greenish or yellowish color that we can see. And then we have what we call the cover color, which is this red coloration that covers many of the cultivars of apples out there. But this is a very important component of quality. Then we also have texture, right? We have here, as you can see in a melon, a very firm melon versus a very melting flesh type melon. We see the same thing with peaches. In the case of apples, we do not want a melting flesh, but we want a crunchier apple, right? So texture is going to vary according within the fruit, the ripening stage in which it is, but also it's going to vary according to different fruits, and we expect different things in terms of texture. Then we have taste. Taste is composed by two different aspects here. We have, first of all, sweetness, which is given by the different contents as well as composition of sugars in the fruits, sucrose, glucose, fructose, etc. And the acid content too. Okay, so it's very important that we find this fine balance between both of them, acids and sugars. It's what is really going to define the taste of fruit, which is a key aspect of quality. And then last but not least, we have aroma volatiles, which are really produced by the fruits, and we sense them through our sense of smell, and also are playing a key role in overall fruit quality. It's important to mention that when we talk about the flavor of fruits, we are really talking about the interaction between taste as well as aroma volatiles of the fruits, okay? So with that said, what we are going to do now is just go into each one of these parameters, and we are going to look into how each one of these is measured specifically, and what is their importance. So let's start with color. As I said, we have that color is going to change as the fruit ripens, as you can see in this sequence of pictures, specifically from plums. And what is really happening in terms of changes of color as the fruit ripens is we are going to see, in most of the cases, a degradation of chlorophyll, which is this disappearance of this green color that you can see here, for example, in the plums. And we are going to start seeing an accumulation of non-photosynthetic pigments, such as anthocyanins, for example, that are related to the appearance of these red or purple colors that we see on some fruits, and carotenoids, which are related to the appearance of these more yellowish or orangish colors that are characteristics of some other type of fruits. Okay, so as I mentioned, we have all these greenish related colors are more what we call the background color of the fruit. And when we are talking about these more red purplish colors, we are talking about the the color that is covering the fruit, right? And they are going to be produced as the fruit ripens. Now, how do we measure color? And here I will be really touching on how do we measure color, or how do we measure each one of these parameters in the lab in order to get a quantitative value, okay? Do not be subjective, but really be able to obtain a value that we can compare with a specific standard or scale, and we know at what point of development or ripening that fruit is, and that will be a good indicator of its quality. So in the case of color, color is measured different ways. Here we are showing you one example, which is the handheld colorimeter. This is the instrument we are talking about, and it is portable, but it has to be connected to a computer generally to make it a little bit easier to work. So what we do is we generally just place the fruit that we are interested in measuring just below the colorimeter. How does this work? Once you pulse a trigger here, there is a pulsed xenon lamp that is equipped with a photocell that is going to be able to detect the amount of light that is passing through a sample. In this case, the sample is going to be our fruit. And this instrument is going to give us a series of measurements, but the one that we are most interested in is what we call the hue angle. This hue angle is expressed in degrees and is what I am showing you here in this diagram. The hue angle is a quantitative value, but it can be translated into a real color. So it will represent the changes in primary colors. When we have a value of 90 degrees, for example, of hue value, we know that the fruit is really yellow. When we have a read around 0 or between 0 and 45, we are moving from a yellow towards an orange towards a red color. When we are around 270 degrees, it's more like a blue color. Okay, 180 degrees, we are talking about a green fruit. So according to the hue value that we are getting, we can directly classify the fruit in a specific coloration, but we also have a quantitative value that we can evaluate further if we want to analyze data, etc. Okay. Then when we want to measure color specifically related to this green color, the background color of the fruit that we want to measure, will be disappearing as the fruit ripens, we can use what we call the difference of absorbance meter, or DA meter. And this is the instrument really. Again, this instrument is very simple to use. You will just place it right next to the fruit you want to measure and the area of the fruit you are interested in measuring. It works by shining LED light to the fruit, and there's a sensor that is going to measure how much light is reflected back out into the fruit, into the instrument. It is going to mainly relate to the content of chlorophyll in the fruit skin. And that's why I'm talking about that this is an important measurement when we are referring more to that background color, to that greenish, yellowish colors of apples, for example, or peaches, when they are still green and they have not ripened completely. Okay. And the unit of measurement here corresponds to the index of absorbance difference, or IID. And the value of IID is going to decrease as the fruit ripens because this chlorophyll is disappearing. So if we think about it, generally when apples, for example, are really green, we can get values of IID around 1.5 or 1.8. And as the fruit starts to ripen, this green color is going to disappear, that we see it, but we cannot quantify it. But with this instrument, we can. And it's going to reach values of 0.2, 0.5. It can even reach values of 0. And that shows us, okay, there's no more green. All the chlorophyll has disappeared. Okay, so again, this is another way of quantitatively being able to measure color of fruits. And both of them are non-destructive, so we are not even, our fruit is totally fine to be consumed after this. So that's about color. Now let's move into our next parameter that is key also for fruit quality, related with textural characteristics. We all know that the softening of fruits is crucial for fruit handling and for the storability potential of fruits, but is also crucial for us as consumers. We want to really enjoy a peach that is soft, that is juicy, right? We do not want to have a peach that is really firm, that has no juice. How are we going to enjoy that? Okay, generally, there can be exceptions. But this softening that happens of the fruits as the fruit ripens on the tree is going to be related with three main aspects. that are taking place there. First of all, cell wall modifications. So we know that plant cells are really composed or surrounded by cell walls. It's our outer layer of the cells. And what is happening is that throughout ripening, this cell wall is going to start to be degraded. It's going to start to be losing its tightness, let's call it. And that allows the fruit to start softening, that we can feel it also with our fingers when we touch a fruit. A second aspect that is related with the softening of fruits is related with turgor pressure. And here, just to make it in simple words, it just relates with the water balance in the fruit. Fruits are generally made up for more than 90% of water. Okay, as fruits lose water because it evaporates, etc., we are going to see that the fruit is going to lose its firmness and it's going to become softer. And but we do not want the fruit to dehydrate, of course. And then the third aspect that is related with fruit softening and fruit texture is the composition of its skin generally, okay? There's fruits that have a lot of lipids or fatty acids in their skin. There's fruits that that would help or not for the exchange of gases in the fruit that is highly related with how this fruit is going to be maintained throughout storage or after it's being harvested. We also have that the skin also can have certain cracks or certain structures that can help also to maintain a firmer texture or not, okay? So these are the three main aspects that physiologically are related with the texture of fruits. And these are all going to be changing in different fruits differently and as the fruit ripens. So how do we measure texture? Generally, texture in fruits is measured in the lab using what you can see in this picture, which is a texture analyzer. It's this big machine here. We position the fruit just below a probe, and we can have more than 50 different types of probes that we can use here, depending on the texture characteristics that we are trying to measure. Generally, if we are talking about fruits like apples, peaches, plums, etc., we are using two main types of probes: a puncture probe, that is the same that you are seeing here, and a compression probe, okay, that is really going to compress the fruit. In terms of if we use one or the other, really, what we are trying to do is to determine the amount of pressure that is required to puncture or to compress the flesh of the fruit in order to get a value, okay? And again, we are trying to quantitatively describe what are the textural characteristics of our fruits. So in the case of the puncture probe, what this will do, as you can see here, it's just going to penetrate the flesh, okay, and give us a value that is determining the amount of force or pressure required to do this. And our result will be transmitted in this type of graph that you see here, where we can we can derive a lot of different parameters from this graph. Like, for example, this highest point in this graph corresponds to the firmness of the fruit, or the area under the curve of this graph corresponds to a parameter related to the work to bite, related with textural characteristics of that fruit, etc., etc. In the case of the compression probe, what this is really doing is emulating how we as humans masticate the fruits. So what the probe is going to do is it's going to compress the fruit, it's going to go down, compress the fruit, then go up, and then compress the fruit again, like when we chew our fruit. And that's why you see two peaks on the graphs here. You see the first compression, then it went down, and then it compressed again, okay? And again, from these same graphs, we can derive a series of texture-related parameters that are going to give us values and are going to help us characterize the texture of our fruit. Another way that we can measure texture is related with using a penetrometer. This is a handheld instrument. We can also use it in the field. It's totally portable. And here it's kind of the same principle. We are just trying to determine the amount of pressure that is required to puncture the flesh of our fruit. Okay, so this is very important because depending on the amount of pressure we need, that is an important indicator to tell us on the maturity stage in which our fruit is. So for example, in apples, when we have 15 pounds of pressure, when we measure with a penetrometer, we know that that fruit is ready to be harvested, okay? When we have 18 pounds of pressure, the fruit is still not ready. So we can still leave it on the tree for a longer period of time, okay? So that's with respect to texture. So now let's move into taste. As I said, taste is composed by sweetness, really sugars, and acidity related to organic acid contents. So in terms of sugars, we have many different sugars that are composing fruit sweetness. We have sucrose, glucose, fructose. We have other sugars that are not mentioned here, like sorbitol, which are sugars that have a less caloric content and a less glycemic index than what sucrose, glucose, or fructose have. So fruits, the composition of these sugars as well as the content of sugars is going to be highly related with the sweetness that we are able to perceive. And the same thing with acids. We have that specific fruits will have characteristic acids that are present in a higher content in those fruits. For example, in citrus, in oranges, we have citric acid. In apples, in peaches, we have malic acid. In grapes, we have tartaric acid. But in general, the main point here is that the taste that we perceive is going to be related to the quantity of these sugars and acids, the composition, which ones are present, and mainly the balance, this fine balance between sugars and acids. When the fruits are very green and they are not ready to harvest, we have a high levels of acids in those fruits and low sugars. So that's why we perceive them so acid. But as the fruits are ripening, acid levels are going to decrease, sugar contents are going to increase, and that's what makes the fruits be perceived to us as highly sweet. Okay. So how can we measure fruit taste? And I will start here by talking a little bit about apples. Apples are pretty particular. They have a lot of starch. Starch is an important carbohydrate that is a component of fruits. And generally what will happen is when apples have high levels of starch, we are going to perceive that they are low in their sweetness level. Because what happens is that the degradation of starch is what is going to be related to the increase in sugars, because starch is going to be converted into sugars as the fruit ripens. So in order to measure the amount of starch, this is a quick measurement. What we use is, and this is used at the commercial level in the industry, we use what is called the starch iodine test. And it's based on this scale. Okay, this scale is showing just how starch is going to disappear from level 1 to level 8. When we have this black-blue coloration is when we have a highest level of starch. And we can see that because this iodine, which is used in this test, is capable of binding to the starch molecules, and they turn them into this blue-black coloration. So when the fruit has a lot of starch and we submit it to this test, this is what we are going to see. But as the fruit ripens, starch is going to disappear, and we end up with a flesh that is totally white. There's no more starch. All the starch is converted into sugars. And this is, you can also see this here throughout these pictures where we are showing how in apples throughout their ripening, you see how starch is disappearing, indicating that there is an increase in sugars. But we can also measure specifically soluble solid contents, okay, or sugars per se. And for that, we use, again, this very portable and simple instrument called the refractometer. What we do here is that we take a juice sample from our fruit and we deposit it here in this lens here, a very small amount of juice. Once we do that, we press start. And this instrument is capable of measuring how the light is going to be bent as it passes through this fruit juice sample. It is going to be able to measure the percentage of soluble solids, which will include mainly sugars, but it also includes a bit of acids and organic salts, etc. But mainly it's composed by measuring sugar content, okay? And there's a ton of studies that have shown that there is a high correlation to the percentage of sugar contents in the fruits by using this refractometer. This is also important. Generally, there are indices of maturity where we know that if a fruit is presenting 12% of soluble solid contents, then that fruit is ready to be harvested. That fruit should be tasting sweet. If we are below that, for a peach, for an apple, that fruit is still not going to be ready. Okay, it still has low sugar contents. And then finally, with respect to acids, that is another important aspect of taste. We can also measure this quantitatively by using a titrator. In this case, I'm showing you the one that we have in the lab, as we have to manage so many samples. We use an automatic titrator where we can place a certain amount of juice from each one of the samples that we want to measure in these tubes. And what is going to happen is that this instrument is going to use a base solution of sodium hydroxide. It's going to add it to our fruit juice until the acids are totally neutralized. And that is going to give us a value that corresponds to the percentage of acidity of that specific acid in that fruit sample. So we can get 0.5% of malic acid, for example, in an apple. That is the type of result we would obtain from this instrument. Okay, so again, being able to quantitatively measure acidity in fruits. And then finally, we also mentioned aroma volatiles, key component of fruit quality too, interacting with sugars and acids in order to define the flavor of fruits. I want to say that the production of volatile organic compounds can be detected by over 650 types of olfactory nerve endings that we have in our nose. So we are really receptive to all of these volatiles. We can perceive them, different volatiles at different intensities, of course. And fruit aroma is going to be influenced by the quantity of these volatiles, but also the composition of these compounds. Okay. So there are some classes of volatiles that directly affect fruit flavor and are widely characterized to be related with a specific aroma kind that we can perceive. So for example, volatiles from the esters class have been known to be directly related with the fruity aroma that we can perceive. Then volatiles from the alcohol class are more related with these earthy type aromas or these more fermented types. Then the aldehyde class with this grassy, bitter, green type aroma when the fruits are immature still. And lactones, for example, are highly related with this peachy-like aroma that we can perceive in peaches or in other fruits. Okay. Now, how do we measure fruit aroma? This is a really challenging measurement to do. We use a technique which as we have so many volatiles that are being produced from one fruit, we need to separate them all and be able to quantify them. So there's this technique which helps to separate them and identify the compounds in their gaseous forms based on their masses. So for that, we use this instrument, which is a gas chromatograph coupled to a mass spectrometry, where we exactly can achieve this. So we can define the volatile profile of our fruits, okay? Instrumentally, and we get again a value of how much of a specific ester is present in that fruit, okay? So that's in general in terms of the different components of fruit quality and how can we measure them instrumentally. But of course, we do not only want to measure this instrumentally, but we also want to perceive this from our senses and be able to evaluate that and to get a value for that, to quantitatively and objectively be able to measure these. How are we as humans perceiving these fruits? It's not just what an instrument tells us, right? And the idea is to be able to correlate the values from the instrumental parameters that we obtain with the values of what we as humans are perceiving. So for that, we use what is called sensory science, okay? And we use two main methodologies. What we do is we can work with what is called consumer testing. What we do here is that we would ask any of you, for example, to please taste a piece of fruit that we want to evaluate, and we would ask you several questions just based on your preferences. It's a very subjective evaluation here. We generally would ask you, okay, can you define the texture of this fruit? And we would give you a scale where 0 is, for example, very firm, until 5 where it is very soft. And you just would tell us what is your perception of this fruit. We could do the same with juiciness, we could do the same with sweetness, etc., with all of these parameters that are composing fruit quality. We generally, if we are using this type of strategy, we would use panelists, or usually we would use more than 100 panelists in order to evaluate the fruits, okay? And the other big methodology that we use when we really want to quantitatively evaluate the human perception of fruits is descriptive analysis. And here what we are doing is we just reduce our group of panelists to 8 to 12 tasters, but these ones have to go through 3 or 4 weeks of previous training. Okay, we are, and this is what you see in these pictures here, really. We have discussion sessions 3 to 4 weeks before they are going to try the actual samples that we want to evaluate, where we all get into an agreement for each one of the attributes that we are going to evaluate. What is really the scale? When we talk about a firm texture peach, if I ask each one of you is going to give me a different definition of what a firm peach is for them and what is a soft peach. But here what we try to do is that everybody is on the same page and they are thinking about the same type of firmness when we are talking about a firm peach, or a medium firm, or a highly firm, or a soft peach, etc., okay? So we only have 8 to 12 tasters in this case, and when they are going to proceed to evaluate the real samples, they do it in booths where they are just individually evaluating their fruit. There's no discussion at all, okay? It's just very focused based on what the training they had before taught them. So this is very important because we are able to get values quantitatively, evaluate what are we as consumers perceiving from these fruit samples. And then it allows us to correlate these values from human perception with our instrumented values for each one of the parameters that compose fruit quality. That's how we can measure it, and we can really define fruit quality in fruits. So with that, I want to move into the second part of the presentation now, where I just wanted to share with you a little bit more of what are the factors that are affecting overall quality, okay? And for that, we have to start by being clear that different fruits are going to be classified differently based on the ripening patterns or the ripening types, okay? And that's where we have the concept of climacteric versus non-climacteric fruits. And this is important because it is going to directly affect the quality of fruits, in which group they are placed. So this is a diagram that shows you some fruits that are classified in the climacteric type and others in the non-climacteric. So when we talk about climacteric fruits, it corresponds to fruits that are able to ripen once they are detached from the tree at the mature stage. So for example, bananas, okay? Bananas are harvested when they are green still, they are not ready to be consumed, but even if we harvest them at that stage and we leave them over the counter, they are going to be able to ripen and to become edible, and we are going to be able to enjoy them, right? That's what the industry does, really. In non-climacteric type of fruits, this does not happen. The fruits cannot ripen after they are detached from the tree at the mature stage. And this is the case, for example, for grapes or cherries or oranges, okay? The fruits have to be harvested when they are ready to be eaten, because even if we leave them over the counter, they are not going to continue ripening. It's not the same case as with bananas, okay? So we have these two big categories, and these are very important because if non-climacteric fruits are not harvested in the right moment, their quality is going to be greatly affected, okay? So what makes this difference between climacteric and non-climacteric fruits? Why some of them can ripen after harvest and why others not? And this all comes back to the fact that we have to remember that fruits are living organisms. Fruits as they are also respiring, okay? And the other big component is a key ripening or hormone that is produced by fruits and by plants, which is called ethylene. Respiration and ethylene are the production of respiration and ethylene are really key in terms of defining if a fruit is being able to ripen after harvest or not. Okay. So when we talk about respiration, we are really talking about the oxidation of the sugars that are present in the fruits to produce carbon dioxide, water vapor, and heat. Okay, as the fruit is harvested, the respiration rate is generally going to increase, and we are going to have an oxidation of these sugars because the fruit has to continue to live, right? And the second aspect is ethylene. Ethylene is this key hormone that is related with ripening, and it's a gas. So it's very important. It's a gas hormone. It's naturally produced by plants, and it stimulates its own biosynthesis. So when fruits or plants start producing ethylene, they are capable, especially fruits, they are capable of producing more and more. Okay, and when we have higher levels of ethylene, we have a higher or an acceleration of the ripening process of fruits. Okay, so let's go back to how does this influence fruits being climacteric or non-climacteric? And that's really what is shown in this graph here. When we are talking about climacteric fruits, the ones that can ripen after being harvested, generally what we see is that after harvesting, we have an increase in respiration, and we also have an increase in ethylene production rate due to this autocatalytic production. Okay, and this is what allows these fruits to be able to ripen after harvest. In the case of non-climacteric fruits, we do not see this increase in respiration after harvest, and ethylene contents are not autocatalytic. They just are maintained low and constant. They never increase. Okay, and this is what does not allow these fruits to be able to ripen when they are just left on the counter, if they are not harvested at the correct stage. Okay. So I just want to quickly go through how do we measure ethylene. Again, we need to quantify ethylene contents in order to know if a fruit is increasing its ethylene or not, and that is going to allow us to determine when we should harvest it or how we should store it. Because if we have more ethylene being produced in the fruit, that ripening process is going to be accelerated, and the fruit is going to be more prone to bruises, it's going to be more prone to diseases, it's going to be more prone to really going to be wasted sooner. So in general, when we are measuring ethylene, as this is a gas, what we do is we just seal fruits in these airtight containers for a certain period of time, and then we collect a 1 ml headspace gas sample, which will be withdrawn from each one of these containers. And as that sample is going to be injected into this instrument called a gas chromatograph, which is going to allow us to quantify the ethylene concentration in our fruit. And based on that, we can make decisions. Do we harvest the fruit? Not still? Are the ethylene levels low? Is this a climacteric fruit or not? Is this a climacteric variety within a fruit or not? Okay, so this is why it's so important to be able to quantify this hormone and understand what's going on throughout the development of the fruit with it. And finally, I just wanted to give you an example of how we commercially, and this is broadly used in the industry, try to regulate the production of ethylene in fruits, and specifically in climacteric type fruits, because climacteric fruits are the ones that can ripen after being harvested because they will have this huge increase in ethylene. But as I said, once the ethylene production is triggered, it will not be stopped, and the fruit is counting its last days of life, let's say. It is going to be more prone to diseases, to bruises. It is going to start a process of just senescing and being less attractive for us as consumers. It's going to start if it's not consumed immediately, okay? So generally, if you think about fruit operations, they are harvesting huge amounts of fruits. And this fruit has to be transported, the fruit has to be stored. So we need a way in how to regulate this ethylene production. If you think about bananas. So for that, there is this chemistry, which is based on a molecule, 1-methylcyclopropane, which is capable of blocking the perception of ethylene. And this is what this picture is showing. Generally, as I say, in the case of bananas, fruits are going to be harvested and ethylene production is going to increase. This ethylene needs to be perceived by certain receptors. But this molecule, 1-MCP, blocks this ethylene to be able to be perceived. So although all this ethylene is being produced, it cannot be perceived, and thus it cannot trigger any downstream signal. That means an acceleration of ripening, loss of quality, etc. Okay, so that's how we can regulate the quality of fruit as well as how long the fruit can last. And here I'm just showing you examples of bananas. And this is bananas that have been harvested and then left at 7 days at room temperature over the counter. And you can see that these are fruits that are not treated with 1-MCP. You can see their status. They are yellow, they ripened, they're ready to eat. And here you can see the same bananas, but they are treated with 1-MCP. You can see how we were able to block ethylene and thus maintain the ripening state so they do not continue ripening. And then we can make them ripen whenever they arrive to the specific market that we want them to be sold in. Here is another example with kiwi fruit. Okay, this is 22 days after harvest. You can see fruits where 1-MCP was applied versus fruits that do not have 1-MCP. Okay, so this is a very important tool because the ability to control ethylene production and thus fruit ripening is a powerful tool to reduce food waste and food loss. We are able to maintain quality, we are able to extend storability, and that helps us to be able to distribute our fruits as we need them to be, and that reduces food waste and food loss enormously. If we just harvested fruit and did not have this type of tool, we would have a lot of fruit that would be just wasted or lost. Okay. So I just wanted to share that example, and so that you also understand the applicability of having this climacteric versus non-climacteric fruit classification. Okay, and that's really what I wanted to share. So as a summary, we learned today that fruit quality is the result of the interaction of different parameters: color, texture, taste, and aroma. We learned that taste and aroma are really what composes fruit flavor. We went through each one of these quality components, and we saw that they can be measured instrumentally, but they can also be measured using sensory science in order to get a quantitative value of how we as consumers are perceiving these fruits. Then we learned that fruits can be classified according to their ripening patterns. We learned about climacteric and non-climacteric fruits, that ethylene and respiration production rates will vary among these climacteric and non-climacteric types. And then we talked about ethylene and how it is important for the fruit industry to be able to regulate these ethylene production rates in order to increase fruit storability, maintain the quality, and reduce fruit loss and fruit waste. Okay. So with that said, I'm just coming to the end of the presentation. I hope this has been useful and that you had enjoyed the information I shared with you. I want to emphasize and highlight that if you have any questions or any comments, please feel free to reach out to me. You can reach by email. We are also actively present in different social media, Twitter, Facebook, Instagram. You are more than welcome to reach out by any of these venues, or follow us if you are interested in the work that we do. So with that, I just want to ask you one favor, if it's possible, if you could please go into this link and rate my teaching if you really enjoyed the talk today, or if you feel that I have to improve something. This is very important for us to maintain getting our funding and be able to continue doing the research we are doing. Okay, so with that said, I really want to thank you all for the time, and please don't hesitate to reach out, and thank you again.